Hello, and thank you for the invitation to join you today and discuss biobanking breakthroughs, innovation, and excellence in Brazil. My name is Wildredo Marin. I am a commercial manager for Thermo Fisher Scientific for the Americas, and I focus very specifically on LN2 cryopreservation and biobanking. So I hope we have a really good discussion afterwards, and then we can spark some curiosity and happy to answer any questions, of course, either with follow up or with the team that's alongside you today. So, for today's agenda, I'd definitely like to make sure that we address at least these three objectives on the screen, meaning to explore the latest innovations in biobanking, and that could include some of the national research initiatives, as well as determine what are the best ways to approach long term biospecimen preservation. I'll talk a little bit about some of the unique best practices that are offered for biobankers, as well as where to find them. And they'll discuss some of the standardization procedures that may speak to your own intentions to maintain quality control and data integrity or regulatory compliance. And lastly, of course, I always want to make sure I offer really good Thermo Fisher Scientific solutions. Great. Well, I've taken myself off camera so that now we can focus on the true lessons for today. And I'm very excited that we're joined by a large group in the audience of biobankers who understand that it was founded on three main pillars. Each one really takes balance and demands very specific attention, but having this social engagement is able to start the conversation so that we can potentially reach more customers in the region and find community resources. My goal would be for biobankers around the world to look to Brazil for one of the great successes in not only sample acquisition, but data driven results. Hopefully, this would lead to some financial success. We could create a strategy to make sure we sustain that growth. And then a lot of my tips offered today will be mainly operational with some equipment solutions, again, optimization of protocols, but also understanding what type of training might be necessary to have a successful biobank. And if you can't already tell, I'm very optimistic about how biobanks can lead in the future. And this starts with the expertise that biobankers offer in the cold chain. We saw this previously during the pandemic, how a lot of leaders sought biobankers to better understand how to preserve biospecimens not only for long-term storage, but also during transportation. There's a mission involved where a lot of this acquisition of samples and usage, utilization is tied to very transparent research goals to help the community. This is a great environment where there are teachers, trainers, and mentors. There may be some opportunity to understand or obtain greater education and degrees. And then most biobankers can develop a career, a long lasting career, because they're working in an infrastructure that may change or adapt, but they understand what's required in order to store these biospecimens, but also help that mission at hand. This will bring together a lot of different disciplines. I know that in my previous biobank, we had biostatisticians, we had clinicians in the hospitals, we also had experts in the different biospecimen materials, such as skin or muscle, that we would obtain. And altogether, you can take action to make sure that, again, we're providing results of these studies back to all of those participants who very thankfully donated not only their time, biospecimen material, as well as associated data. So, an additional reason why I'm very optimistic is because in my current role, I do have access to market reports, and I can tell you that biobanking will be growing, and it'll be driven by the need and a demand for high quality samples. And this means that these samples will have to come with associated data. Usually this is de-identified or anonymized, but is this actual data that accompanies the biomaterial will be what provides the most value and will translate to those scientific discoveries. We know that we can improve the process of obtaining and organizing our samples with automation. This will help minimize the human touch points and create less errors. We also know that there will be a need for national um, groups and institutes to work together, potentially influencing government to obtain better funding, and of course, make sure that um, there's at least one population program per region. And then, of course, a need for clinical trials and in hospital sectors where we are trending closer to precision medicine type of discovery, which will provide some broad information about the genomic characteristics of a particular region, or again, move towards curing a very specific disease within individuals or what's highly prevalent again within the country. So I'll use precision medicine as an example of how biobanks can support these type of initiatives. And they do need not only clinical partners, but it is usually a national effort that involves several community networks. So, for example, I worked in a juvenile dermatomyositis or pediatric rare disease biobank, but we worked with a local children's hospital, and those clinicians and those physicians would really help us identify which patients we could enroll into our study. Um, we'd like to make sure that that group or that cohort would be diverse and inclusive, children of all ages and races. But of course, we would want to make sure that we had proper controls whenever we were working within these networks. We also sought to have high sample volumes. So we wanted to make sure we can get them at different time points. That means you want longitudinal cohorts. To some people, that means that yes, you are going to accumulate a lot of samples, but the time points is really what's going to matter. And then, of course, being able to organize all of those samples will require knowledge of cold storage infrastructure. You may also want to share some, which is a terrific part of biobanking, just this collaboration and interoperability. And those that you're not sharing, of course, you're going to want to use in a lot of the omic analyses. So that includes genomic sequencing. We did that with qPCR samples, again, obtaining DNA or RNA. Anything that we had for serum or plasma, we would use for proteomics. And then, or at least quantification of some of the proteomics using those longitudinal samples. And then, of course, biomarkers are a big part of most biobanks where you can obtain some genomic profiles. Usually for us, we would try to get them prior to diagnosis, during treatment, and then see what their response was to the treatment. And yes, I do feel very strongly that biobanks in Latin America have the foundations to develop the way scientific research of infectious diseases should move forward. We know that that's a clear advantage in Latin America, and it starts by understanding what are the local resources, potential pitfalls, but taking those and turning them into very sustainable strengths. And the examples below being that Brazil is home to the fourth biggest city in the world, right? It's an increasing number of inhabitants in high density areas. Sometimes they're very poor, but in other cases, this could mean a very diverse population cohort for enrollment in very specific studies, and that leads to greater trends and observations for the epidemiologists to be able to publish. Similarly, there's access to various biomes. We know this might accelerate pathogen transmission, but it also aids the agricultural conservation and industrial stability. I know I saw a very interesting presentation several years ago now by Embrapa during the Biobank conference and how their template can be expanded to countries. I also get asked questions about microbes in the soil a lot. And when I think of access or I think of transportation roads, I think of much needed infrastructure to get vaccines or samples in and out of all the various states, and make sure that those states have enough access and understanding of the proper storage temperatures. Lastly, there's obviously a growing need in healthcare, and there's an opportunity for leadership there. And this means creating increased research infrastructure and visibility and collaboration. And of course, much of this will depend on having additional global knowledge and symposiums to address this and promote access and mobility. So very quickly, another way to help approach best practices, just like in the previous slide where we're talking about understanding potential gaps and turning them into strengths, you have to internally audit your process. I mean, I showed this slide at a lot of meetings because it's basically the top three or maybe the first three things any biobank should be doing is understanding and establishing their protocols by going to the proper best practices or references of best practices. Of course, ISBR and the National Cancer Institute are great. And of course, there's offerings for QBRS in terms of biorepository science certification. Every biobank should develop an emergency response plan, being able to pivot or understand how to save or move samples in case there's some geopolitical event or natural disaster. Of course, we want to make sure everything is backed up and properly stored. And one of the biggest things that will be looked towards the future expectation or requirement of all biobank is having accuracy of data records. So just knowing who has access to them, who's in charge of them, and how they can be properly not just shared but used as part of the research investigation process to make sure that the samples are properly de-identified. I like this slide because this is something we know very well, how to store samples in a biobank. We can offer some of the best guidelines and recommendations for long-term storage. Of course, with that first example, when we think of temperatures for all the different sample types in a biobank, I like to think of DNA as a really good example because usually we can isolate that from cells that are stored in a cryogenic environment, but the DNA itself is stable at -20, so that might be just a freezer type of piece of equipment versus something that requires ultra low temperatures and RNA, which is closer to -80 because it's less stable material, but equally important information that you can get from mRNA. Again, we try to offer guidelines how to avoid expired consumables or what temperatures your consumables and media should be at. We want to make sure that you're able to organize them and add value to any legacy collections by following a quality management program. Of course, if we can provide you with the proper accessories and labeling, you're more confident, and the biobank will hopefully be more successful and able to operate with others and collaborate with others equally successfully. We try to engage you and engage your staff, not only to offer opportunities where they can basically create a career in biobanking, but also understand that knowledge and use of the equipment will help maximize the production in a facility. So one of the questions we get quite often is whether there's a need or a requirement to follow ISO 20387 to help standardize the processes. And this is because it's coming from the International Standard Organization, and it's a general requirement for biobanking. It's applicable to all organizations performing the biobanking activities, which is sample acquisition, storage, as well as analytics. And this belongs not just to research, but also some of the clinical work I mentioned earlier. This is discussing a requirement to provide the proper resources, which means personnel in a facility, make sure they're properly prepared, as well as have protocols that are written and standardized. And the samples are preserved as they should be, again, in those optimal temperatures I mentioned, and there may be a quality control process to make sure that the value of some of that associated data is captured within each one of the samples. All this is very well documented through acquisition process, any type of transportation, and of course, the short-term and long-term storage. I know that this one's being currently reviewed. I think it's in the committee, so there'll be some updates later. But we do see a trend of, because it does require some initial funding, we do see some trends that it's increasing in popularity as a way to work closely with partners in industry. If a biobank in academia can actually follow some of these requirements. So let's get to some solutions, and we'll start rethinking the ULT, ultra low temperature freezers. So I'm really excited because I get to show you a slide that we shared at the ISBER 2025 meeting in Montreal earlier this year. Of course, we did focus on these three functions of Thermo Fisher Scientific, not just the equipment here with the universal ULT freezers, which was launched last year in April, again at an ISBER meeting and in Analytica in Germany, to great reviews. We discussed a lot of these questions, and of course, I encourage you, if you're interested, we can talk about that in the Q&A later today, or ask my colleagues, or actually see if we can access the recordings from that ISBER meeting where we did get to talk about this in depth. We also talked about the consumables, best ways to manage your samples, and of course, we talked about some of the biorepository options in case you look to scale your biobank beyond your own building facility. One of the reasons that we discussed the TSX Universal Freezers because we're extremely proud of the approach and the impact that it's having on the environment. Basically, allowing users to reduce their energy footprint because they're so energy efficient. We're very happy that a lot of the participants in the freezer challenge last year actually were customers of Thermo Fisher Scientific, and combined that challenge alone, along with other manufacturers, saved almost 32 million kilowatt hours and 2,200 metric tons of carbon dioxide. And so, again, knowing that we are introducing a product that is able to contribute to this type of, to meet the challenge of reducing emissions is very important to us. And I'd like to discuss that more and show you why. So internally, Thermo Fisher has its very almost stringent requirements as an approach to design for sustainability. We're trying to discuss the impact areas. Every product should at least have one or two of these impact areas that's coming out or being developed, meaning less hazardous, less waste, more efficient, energy efficient, and responsibly packaged, and an extended lifespan. And these impact areas are taken into consideration of the design of the product life cycle. So anything that you're starting to see from Thermo Fisher Scientific that is a greener by design should have obviously incorporated some of the impact areas. So very quickly with this greener by design, you'll start to see that a lot of the Thermo Fisher Scientific, at least over 5,000 of our Thermo Fisher Scientific brand, will have some of this green leaf because they meet those areas. And then the Fisher Scientific also uses a greener choice badge on over 60,000 of the products. Again, we're very happy that we're able to partner with some of these third-party eco-labels. That includes the US EPA. So that's Energy Star 2.0. So it's a lot more stringent in some of the measurements across the different cabinet sizes for kilowatt hours. We have over 130 products from Thermo Fisher Scientific that meet Energy Star requirements. We also have My Green Lab ACT label, which stands for accountability, consistency, and transparency. And we like to think of it as like a eco-nutrition label for some of the lab products, where a lot of the scores are listed on there depending on what are the needs of a user. And we have one of the biggest catalogs for the My Green Lab ACT label, at least over 355 now as of today, actually. We also partner with ISCC to get certification and make sure we properly have identified and have traceability to chain of custody for some of the recycled content materials. So again, going back to those impact areas and how they apply specifically to the TSX Universal, well, you'll see that now all the refrigerants in the unit have a GWP, or global warming potential, of 0. They are made with less waste, and they're made in a facility. It's a zero waste to landfill facility with 100% renewable energy in Asheville, North Carolina. I mentioned they come with the Energy Star 2.0 certification. They are responsibly packaged, so they are robust. They can make it all around the world safely, but of course not burdened with heavy type of material that's just not responsible or excessive. And then of course our confidence in the units themselves is expressed with this 12-year warranty, which is probably the leading one in the industry, significantly longer than other products, other ULT freezers. So this is another great slide that we showed at the ISPER meeting, and this is one that really stood out to the audience, because what we're basically saying is that we know biobankers use a lot of freezers, but we are able to introduce or hold much more capacity with less freezers from Thermo Fisher Scientific, this TSX 700, compared to brand P and their 25 cubic foot. You would be able to get 2 less freezers overall. And so what does that mean? It means, well, it costs less. It costs less not only to qualify and install them and maintain them, but with each one of these, you're getting not only additional capacity because there's 700 box units, but you're getting a faster door opening recovery time with the unit, meaning you can go into it maybe within 15 to 20 seconds after opening the door for 1 minute. There's tighter peak variation, and it takes longer for them to warm up after the door is open. Even starting a freezer, if you were to use it as a backup, the smallest cabinet one, I believe is maybe a 400, would probably reach -80 within 3.5 hours if you were in very quick need of a backup freezer to put in samples. So here we have a really nice picture of the entire ultra-low temperature freezer family with 2 upright versions, the TSX Universal freezers in the middle with the light bar accessory and a lot of the eco labeling. We also have a chest freezer off to the right where you might get some additional efficiency because of course cold likes to stay low. And when you open these doors, organization and formatting of the samples is really efficient. If you're thinking about scaling, these are some very quick points about making sure you get organized and again, follow the proper, not just requirements, but best practices to standardize your process. Make sure you're able to track your samples, organize them in your freezer, and then make sure that if you're able to minimize those human touch points with automation, include that in your process. I know that it's easier for some to make small improvements, but in some cases it makes a lot of sense to just start fresh with a new method, basically incorporate it and not just standardize it, but just have a cutoff point where you started to process these samples in this manner from that point moving forward. And make sure you share and track all your results. We'd love to hear that, and we want to make sure we can help. Okay, so now sometimes you've outgrown your facility and you're looking for a partner. So I'll go very quickly through these slides just to tell you some of the options in our biorepository teams. Um, some reasons to outsource the project is because you just want to avoid paying for everything yourself or leasing units. Of course, we have facilities that have them ready to go, and it's just a matter of understanding what your needs are. When you want to simplify things, of course, the only difference is that instead of storing them on site, you're storing them off site, but you're making sure that you can focus on what matters, whether that's the analytical equipment instead of the machinery for storage. You obviously have our expertise, and we have very strict compliance. And then, of course, any of the risk is assumed by the facility because we're trying to make sure we're helping you, and we have proper staff and monitoring to make sure that everything is kept as securely as possible because we know how important it is to you and the community. So just again, another quick points about inventory management and what we can do to help to go from cold to cryo across some of our facilities globally. Here's a quick map of where they're located, and then obviously I'd like to highlight, yes, we do have some, we have one in Brazil and Sao Paulo. And then as the LN2 commercial manager, I'd really want to make sure I hit on these points in terms of cryopreservation equipment. I love the flexibility of the different sizes of this Cryo Plus. It's easy access. It's a large opening up top. You can use it with vapor phase or liquid phase, and they're autofills, as is the Cryo Extra, very popular in our APAC regions because of the large high efficiency and capacity of the unit, as well as the controller. You know, for smaller labs, we see a lot of people use locators. We can put these on carts. And then we have a lot of shippers like this, which we can put, we can track with like a smart tracker, but then you could also put them in this type of hard shell and ship them across the country. If you'd like to learn more about additional scaling considerations for higher throughput, I will make the slides available, but you can see some of the options I've discussed at some of the biobanking meetings. Similarly, when I talk about sustainable sample analysis, we have some terrific instruments, and of course, reach out to our teams nearby to discuss the Ion Torrent Genexus system. But of course, without the additional support from our Genomic Sciences division, we wouldn't be able to sponsor the event today. And so I'm very happy to share at least some quick bullet points on the microarray scanning or the larger high throughput scanning with the GeneTitan multi-channel instruments. We want to hear about all of your successes and how we can help. We have a terrific biobanking hub that's gotten some recent updates, so you'll see some videos and some blogs. Of course, access to some of our own best practices for the equipment, so please make sure you look towards thermofisher.com/biobanking. And so with a minute to go, I guess I could just put everything out on this slide and leave it up for a little bit. But again, thank you very much for the invitations. If you have any questions, please feel free to follow up with me or our team that's present there. Of course, I'd love to connect with as many of you as possible and definitely enjoy the rest of the meeting.